Let's go ahead and begin where we left off last time. Last week, we were talking about the reticular theory, which was posited by Golgi. So Camillo Golgi was the one who talked about the reticular theory. And basically, the whole theory was just, in a nutshell, kind of envisioning the neuronal system as all working continuously. And you could sort of think of it as the way the heart works. Right, the way the cardiac myocytes all come in contact with each other, it's through gap junctions, for example. You have your intercalated discs where you have gap junctions where calcium, for example, can flow through freely from cell to cell. So Golgi's theory of the neuronal system was kind of similar to that. It was kind of how all the tissues kind of work continuously together, and that there wasn't like a single discrete individual cell that was actually doing one thing to, allow for downstream communication with other cells in the neural system, but they all just kind of worked as one. Whereas with Santiago Ramon Cajal, his theory of the neuronal doctrine posited that cells within the neural system each had a discrete and distinct function, and that the cells were all kind of interconnected through, synaptic type connections. And so his theory was that the neurons were the basic signaling unit of the nervous system, separated and working as separate, distinct, and discrete cells. So that was the summation of Cajal's neuronal doctrine. But I will say, though, that Camillo Golgi was one of the major figures within the early development of neurology. Golgi, of course, is the one that came up with Golgi staining, which was used heavily by Cajal. So if you notice over here, this image in the bottom right corner, that was one of Cajal's very first illustrations of neurons. And so Golgi staining is effectively using silver stains, so silver salts, so that you can stain all the neurons. And as I mentioned last week, back in the 19th century, they didn't have cameras to be able to take a photograph of what they were looking at underneath a light microscope. And so Cajal was doing all the diagrams and illustrations by hand. So these are all hand-drawn illustrations. Cajal was also considered to be an artist at the time, and so his contributions were to be able to illustrate what a neuron and neuronal systems looked like very early on in neuroscience. So super cool dude. His findings were, of course, later verified through electron microscopy. So make sure you keep a note of that. Electron microscopy was the way that the neuron doctrine was actually solidified as like the governing theory of the way neurons are able to communicate with each other. So that kind of dispelled with the whole reticular theory posited by Camillo Golgi. So here's another image of neurons as they work in systems together, kind of like further illustrating how the neuronal doctrine operates. So here's an image of the olfactory bulb. So over here in A, you got the olfactory bulb. B is where you have all of the actual receptors within the nasal cavity that are going to be, chemoreceptors that are receiving sensory input from different types of scents, for example. Those signals are going to be sent over to the olfactory bulb, and then from the olfactory bulb, it's going to be sent off into the cerebral cortex. And recall, cranial nerve 1 does not go through the thalamus, and so these neurons would go straight off to the cortex to be able to get integrated into, for example, memory, so that you have memory of different scents. Here's another illustration that was produced by Santiago Cajal. So this illustration is demonstrating basically glial cells. Glial cells, again, those are going to be all the supporting cells of neurons. So you have your neurons, right? That's going to contain things like dendrites, the soma, the cell body, axons, and then you have your glial cells. And the glial cells are all the cells that are going to be providing supportive functions. So you have Well, let me ask you a couple of examples. So you guys tell me, what are a few different types of glial cells? I personally hate doing lectures over Zoom because it kind of takes away from like the interaction between you and me. So, it'd be fun to at least get a little bit of that kind of interaction. So give me a couple examples of what would constitute as glial cells.
Is an astrocyte a glial cell?
Perfect. What do astrocytes do?
Blood-brain barrier?
Yeah, absolutely. That's perfect. What's another example? Someone else. I'm going to start calling names here. Kezia. I hope I pronounced your name correct. Give me another example of a glial cell.
Oligodendrocytes.
Beautiful. What do those do?
The main isn't it the main glial cell type? It makes up the sheath.
Yes, beautiful. The myelin sheath. Now, for oligodendrocytes, is that within the central nervous system or the peripheral nervous system?
Did you say peripheral last time? Or is it central?
Central, yeah, central nervous system. So oligodendrocytes provide myelin sheaths for the central nervous system. All right, who else here? Tamara, what is the cell type that provides myelin sheaths for the peripheral nervous system?
Schwann cells.
Schwann cells, awesome. All right, now I'm going to call on somebody else here. Israel, what is another type of glial cell that is going to be involved in gobbling up debris or even like perhaps foreign invaders or things like that?
Would microglia be?
Yes, beautiful, exactly. Microglia, that's perfect. So those are some of the major types of glial cells that we're going to find within the central nervous system. And so here's an illustration that Dr. Cajal did of a lot of those different types of glial cells back in the 19th century. Another thing to keep in mind too about neurons is that they're going to be polarized. So they're going to be polarized, meaning that you're going to have, for example, on one end of the cell, you're going to potentially see things like dendrites as well as the cell body. And on the other end of the cell is going to be the axons. And so you have polarization. So you have a receiving end, that's going to be your dendrites, right? These are going to be your receivers, right? And then you have your transmitting end, and that's going to be your axons. And those are what transmit signals out either to subsequent neurons through synaptic connections or at the neuromuscular junction, right? At the end plate of the neuromuscular junction. So you're going to be sending off acetylcholine, for example, that attaches to nicotinic receptors to allow for muscular contraction. So here's a couple images from your textbook. Not super high yield for the sake of your upcoming exam, but just to kind of further demonstrate different types of imaging techniques. These are using, for example, electron microscopy. So these are transmission electron microscopy images. So electron microscopy, there's a couple different types. You have scanning electron microscopy, where you can actually see like the surrounding, like the outside layers of whatever it is that you're looking at, right? For this, for the sake of neurons, we'd be looking at what the outside structures of the neurons look like. Whereas with transmission electron microscopy, that's what you're seeing here with this type of image. Transmission electron microscopy, you can see right through the cell, and you can see all the different structures involved. So for these images here, you're going to see myelin sheaths on axons. For example, over here, that's going to be in this kind of like darker type color. The gold also here is making up those myelin sheaths that you can see on these images. So those are nice little like transverse type cuts through an axon. Over here you see the soma, or the cell body. You can see the nucleus. That's where all the transcription is taking place, where you're turning DNA into RNA, and then eventually you're going to be turning that RNA into proteins through translation. And so all that's going to be taking place within the soma. You also have lots of Golgi apparatus. You got your rough endoplasmic reticulum, endoplasmic reticulum, ribosomes. That's where you get all the protein synthesis within the cell body. And eventually, whatever is being synthesized within the cell body gets transmitted all the way down the length of the axon using microtubules. And so later when we start talking about, for example, neuronal communication, we're going to be talking about microtubules and how you have things like neurotransmitters being sent all the way down to the axon terminal so that it can be sent off into the to stimulate adjacent cells. And so here's another image here. You got your oligodendrocyte. So that's what this gold image over here, those are the actual cells that are able to produce the myelin sheaths. When it comes to oligodendrocytes versus Schwann cells, what's one of the major... I think someone's got their microphone on. What's one of the major differences between oligodendrocytes and Schwann cells, aside from the fact that oligodendrocytes are in the central nervous system versus Schwann cells, which are in the peripheral nervous system? Do you guys recall from last time? It has to do with the number of neurons that are myelinated.
Are oligos just one per cell?
So, no, it's going to be the opposite.
Oh, okay.
So within the central nervous system, the oligodendrocytes are able to myelinate multiple different axons. Whereas within the peripheral nervous system, it's going to be 1-to-1. It's going to be one Schwann cell per axon. So that's going to be one of the major differences between those two. So here is what's going on in both the soma as well as within the axon. So again, protein synthesis, that's going to be taking place within the cell body, right? Whereas within the axons, that's where you have the transmission of things like neurotransmitters, right? And so I haven't done a Zoom lecture in a long time, so let me see how I can switch over to the the video here. So hopefully I'm able to screen share the video so we can watch that together. So just bear with me a second here. Hold on 1 second. I might have to stop sharing with the PowerPoint and then reshare with the video. So hold on 1 second. Let me reshare. All right. Do you guys see my web browser right now? Do you guys see my YouTube page here?
Yeah.
Yes.
Okay, cool. So here's one image of transmission of different contents through the axon. So this is a really cool image. They used fluorescence to be able to light up the different things that are going to be moving back and forth on the axon. So this is just one giant axon. Let me replay that again. Dang it. So super cool image. You can actually see things moving either away from the cell body or back towards the cell body. And the way that this is done is going to be through different types of proteins. So you have dynein as well as kinesin. So let me play another video that demonstrates how kinesin works. Do you guys still see my web browser here? Let me stop that video. So here's an image of kinesin walking. So it's literally a protein that basically walks along the microtubule. And the microtubules are going to go all the way from the cell body towards the axon terminal. And so that's how things like neurotransmitters, for example, that are synthesized within the cell body get transmitted all the way towards the very end of the axon terminal. So super cool stuff. And so dynein, that's going to be more associated with retrograde transport. And so dynein is what brings things back towards the cell body, whereas kinesin is going to be what brings things from the cell body towards the end of the axon terminal. And again,
your slides.
oh, sorry about that.
It's okay.
Thanks for letting me know. All right. So again, like I said, kinesin is going to be walking towards the axon terminal, whereas dynein goes back towards the cell body from the axon terminal. And then again, they're polarized in terms of like the cell, the cell structure, cell shape. So you have your receiving end versus the end that's going to be putting out different types of signals from the axon. The postsynaptic cell, that's going to be the receiver. So you have your presynaptic cell, and we're going to talk about this when we get into synaptic transmission. So you have your presynaptic cell. That is what the cell, that's going to be the cell that's going to be sending out information. And then when you have the synapse, you have the postsynaptic cell. And the postsynaptic terminal is where you have, for example, receptors that are going to be receiving signals from the neurotransmitters being released by the presynaptic cell. Two major terms that I want you guys to be familiar with as well. Pretty straightforward. Divergence versus convergence. With divergence, that's where you have one cell being able to communicate with multiple postsynaptic cells. So you have divergence where you have one cell communicating with multiple postsynaptic cells versus convergence where you have multiple cells communicating with one single neuron. And so this illustration over here demonstrates that those two different concepts pretty well. So convergence, I don't know if you guys studied evolution, but similar terms are also used in evolution where you have convergent evolution versus divergent evolution. Similar type of concept when it comes to convergence and divergence and neurology and the way that cells are able to communicate with each other. So here are your glial cells. We kind of already talked about these already, but just to reiterate, astrocytes, those are going to be what allow for the formation of the blood-brain barrier. Oligodendrocytes are going to be what you find within the central nervous system. And again, those also provide myelination from multiple different axons, not just one. And then microglia, those are going to be your macrophages of the central nervous system. Those are what are able to, gobble up either foreign invaders. If you have any sort of injury to the brain, for example, like a stroke or something like that, where you have debris, for example, like blood, right? If you have like a hemorrhage and blood spills out into the central nervous system, over time, microglia are going to be able to gobble up all those, all that debris and fragments over time, so it can help to clear things out. Also, microglia play a really huge, important role in clearing out misfolded proteins, things that accumulate in diseases such as Parkinson's disease, Alzheimer's disease, as well as Lewy body dementia. We're going to talk about those types of proteins later on. But for Alzheimer's, you have neurofibrillary tangles and beta amyloid plaques. Those build up over time, and that's what causes the damage to neurons in the brain. For Parkinson's disease, you have alpha-synuclein accumulation. Those are misfolded proteins as well. And then Lewy body dementia is also going to be involving both. It's a combination of Alzheimer's and Parkinson's with Lewy body dementia. So you get alpha-synuclein accumulation and you get beta amyloid plaques. But in normal individuals, those type of misfolded proteins are going to be gobbled up by microglia. So let me play a little video real quick showing the cell motility. Let me share my browser with you guys. If this starts to get too tedious in terms of like switching back and forth, I might stop doing that. But let me play this video real quick. So here's cell motility. So this is microglia. So microglia kind of have like these little appendages that kind of, scour the environment to look for different types of things that are that need to be cleared out. And so you can see the microglia over here moving about the environment, looking for things to gobble up. So that's a cool little video. Let me play another one here that talks about plasticity of cells. Are you guys still seeing my browser here with the YouTube?
Good afternoon, everyone. I would like to welcome you.
Yeah, let me stop that video real quick here. All right. So
microglia and neuropathic pain plasticity. Glial cells react to stimuli that
I
threaten
can't see it.
homeostasis in the central
We
nervous
hear the
system.
audio.
You only hear the audio? All right. That's a huge bummer. All right. So that being said, I think maybe just to avoid switching back and forth between different, screens, I'm just going to go through the lecture slides in that case. And the links, they're embedded within the PowerPoint presentation, so you guys will be able to have access to those. You can watch those on your own time. So my apologies. Do you guys see my PowerPoint presentation?
Yeah.
Okay, cool.
I have a question.
Yes.
So with the microglia cells, is there like a way where they work better? Like I've heard like when you're fasting, how certain cells can regenerate and you can increase stem cell production. But I was wondering if that would have the same effect with your microglia as well.
Oh yeah, that absolutely is the case. So here's something that is really important in normal physiology. It's sleep, right? I think, did we talk about this already in this class where you have to have at least
regenerates when you're sleeping?
Exactly. Yeah, you have to have a certain number of hours of sleep. There's been studies that show, that, for example, individuals that have compromised sleep over a long period of time tend to have higher rates of, for example, Alzheimer's, Parkinson's disease, right? Two things that we just talked about a second ago. And why is that? Because during deep sleep, during REM sleep, that's when you have higher activity of microglia. And so during your deep sleep, that's when microglia are able to actually go in there and start scouring the environment within the central nervous system to be able to start clearing out things that accumulate throughout the day. And so, yeah, absolutely. Lifestyle choices, diet, exercise, things like that can all have impacts on your immune system. And you can think of the microglia as being sort of part of the immune system of the central nervous system. And so sleep is really important for that. And I would imagine that other things like diet and exercise would also have implications on the function of microglia. So yeah, that's a really, really good question. Yeah, absolutely. So here's another image. This is just from your textbook talking about the different types of cells. So you got your astrocytes, you got your oligodendrocytes, which provide the myelination for all the different axons within the central nervous system, and then your microglial cells that basically act as phagocytes of the central nervous system. And then in your textbook, they go really heavily into the different types of staining techniques, which, the main staining techniques that I want you guys to be familiar with would be your fluorescent stains. So here in G, for example, you got some antibodies that are going to be used in fluorescent stains. The other types of staining that we're going to talk about a little bit later are going to be your Nissl stains, your crystal violet stains, as well as your Golgi staining. So your textbook definitely goes into a lot of detail regarding staining techniques. So here's your stains that I'm going to definitely test you on. So make sure that you are familiar with these four major types of stains. So Golgi stains, those are the ones that were pioneered by Camillo Golgi, again, used also by Santiago Ramon y Cajal when he was doing all of his little illustrations of neurons. What do Golgi stains use? It's basically silver salts. And when you use a Golgi stain, those cells are going to turn basically black, and you can visualize those underneath light microscopy, and then be able to illustrate them from there. Fluorescent dyes, basically you have a label, right? So you have labeling of different regions of the cell, for example. You can stain different types of proteins, use specific type of probes, for example, and those are going to light up. That's basically it. So that's how fluorescent dyes work. And then Nissl staining, effectively you're staining things like DNA, RNA, anything that contains like nucleic acids. And so they're going to be negatively charged molecules. That's what the Nissl stains target. Anything that's negatively charged, and RNA and DNA are going to be negatively charged. and they're going to stain basophilic. And whenever you see something that's basophilic, it's going to stain blue. And so the top right image over here, that's an example of your Nissl stains. And then finally, crystal violet. Crystal violet is a compound that basically stains in a type of purplish color. Pretty straightforward. So make sure that you're familiar with, if you see an image of different, neuronal tissue that's stained with different methods, make sure you're familiar with what those staining methods probably were. So here's several examples of Golgi stains. These are all done with your silver salts, so all the neurons look black. C over here is the only one that's going to be stained using fluorescent dyes. And then here's a couple of other images of Nissl versus crystal violet stains. So E over here is going to be a crystal violet stain. Same thing with F. G over here is going to be using a Nissl stain. H is also going to be using a Nissl type stain. All right, any questions on staining techniques before we move on?
Really quick for the crystal violet stain.
Yes.
I know we said it was purple. What was the other thing to keep in mind with how it operates?
It just allows you to see different layers. Let me see a good example here. So crystal violet was used in E. So in here you could see, I don't know. You could see like different areas that were stained with the crystal violet. You can see the dendrites. The axons are not labeled here. So anything that's like not labeled here is going to be in blank. But it's kind of hard to see the difference between crystal violet on E versus H, because H is going to be a Nissl stain versus E using crystal violet.
So basically the main takeaway is that the Nissl stain is negatively charged DNA, RNA, and another one is
mostly staining, mostly staining anything that's negatively charged, versus a crystal violet stain is just going to be entering into the cell. So it's going to be kind of similar to when you're doing like gram staining in microbiology, where you have the stain just entering into the cell. So if you look at, like, for example, like methylene blue, it basically just enters into the cell and you can see the entire cell. Versus with Nissl staining, you're specifically targeting things that are negatively charged, like your DNA and RNA. So that's kind of like the major key takeaways between crystal violet versus Nissl stainings.
That definitely helps. Thank you.
All right, cool. All right, neurophils and neural circuits. A neurophil is going to be something that you find containing lots of glia as well as neuronal tissues. So it doesn't have much in terms of cell bodies. So neurophil, when you contrast that with gray matter is going to be higher in like cell body content. So you're going to have lots of dendrites, lots of cell bodies, versus a neurophil is specifically going to be glial processes, but very small number of cell bodies. You see a lot of synapses though, but not much in terms of cell bodies. So it's going to be glia plus axons plus synaptic terminals. And so here's an image from your cerebral cortex. You have several different layers within the cerebral cortex. And within different layers within the cerebral cortex, you're going to find lots of neuropils. So fewer cell bodies and more in terms of like synapses, axons, glia. And then we already talked quite a bit about afferent versus efferent signals, but anything that's afferent is going to be signals that come into the CNS, right? It's going to be coming from the peripheral nervous system, sensory input, and then it gets integrated into the CNS. And then anything that's output is going to be efferent signals. And efferent can be anything from excitatory, inhibitory, modulatory type signals. And we're going to go into all those later on in the next couple weeks. And then interneurons, those are going to be really, really short neurons that you find, especially within the spinal cord. And they're going to be involved in like local circuit type functions. especially involved in, for example, your reflexes. So we're going to talk about all sorts of different reflexes later on this semester, but first we're going to do a little intro on the myotactic type reflex. And so what's going to be involved in a reflex? You're going to have four major things. So these are the four. So first you're going to have your sensory input, okay? And that's going to be an afferent signal. So let's use the patella reflex as an example. So when you take your reflex hammer and then you hit the patella tendon, that's going to send afferent signals back into your dorsal column to your spinal cord. Then you're going to also have interneurons that are going to serve basically as a middleman. They're going to help to basically regulate the signals either to turn on or turn off different neurons. And so for the myotactic reflex specifically that we're going to talk about in a second, the interneurons are going to serve as inhibitory type neurons. They're going to inhibit the flexor muscles of the thigh, which we're going to talk about in a second. And then you have both your extensor and your flexor muscles. Extensor muscles for the patella reflex, those are going to be the ones that get activated, versus the flexor muscles are going to be the ones that are inactivated. And they're going to be inactivated specifically by interneurons. So interneurons are going to send inhibitory signals to your flexor muscles. And so here's your myotactic reflex. We're going to, again, go through all these later on. I'm going to go through every single different type of reflex that you would do in a neurological exam on a patient. And so we'll bring in some reflex hammers for that. But in this image over here, you see on the patient's left side, he's got a normal patella reflex. So that's what you would normally see. Versus on the right side over here, you're seeing exaggerated patella reflex. Now, if you were to, take a guess as to what kind of lesion this patient might have had, what do you think this patient had? Was it an upper or a lower motor neuron lesion? And on which side of the body?
I'm going to say on the left side, and I'm going to say upper motor.
Perfect. Yeah. Why is it an upper motor neuron lesion?
I will say why I thought it was left side, because we said it was bilateral or opposites.
Well, it would be contralateral. Yeah, contralateral. Yep.
And upper motor, because motor happens in the frontal area.
No. So what's going on over here? So this person over here has a normal reflex on his left knee, versus the right patella. He looks like he's got a little bit of what? Hyperreflexia. And so hyperreflexia usually indicates what type of motor neuron lesion, upper or lower motor neurons.
Lower. Is it lower?
So when you think lower motor neuron lesion, I want you to think that everything basically goes down. So with a lower motor neuron lesion, anything that's like outside of the spinal cord, say for example, the nerves that innervate your flexor and extensor muscles in the thigh, as an example. If you were to sever those nerves, what do you think would look like if you did a patella reflex on that patient? What do you think would happen to their leg? Do you think anything would happen really?
It wouldn't move.
Yeah, it wouldn't move. They would be basically flaccid. So if you have a lower motor neuron lesion outside of the spinal cord or the central nervous system, then that would result in hyporeflexia or flaccid paralysis. Versus a patient with an upper motor neuron lesion, they're going to have hyperreflexia, or they're going to have more of a rigid type paralysis, right? They might have tetany as an example. So this patient probably has a left-sided upper motor neuron lesion, probably due to a stroke in their brain.
Could you please explain why again?
Okay, so upper motor neuron lesions, you get hyperreflexia. I don't know exactly why you get hyperreflexia with an upper motor neuron lesion. With a lower motor neuron lesion, it makes more sense because if you sever the nerve's not going to be able to have any sort of
movement, right?
Yeah, you're not going to have any movement whatsoever. But why do you get hyperreflexia with the opposite? That's actually a really good question that I'm going to have to actually look into. So let me make a note of that. And I will get back to you on that later when we meet again on Thursday, because that's a really good question. So you caught me off guard. Just give me 1 second. Let me make a note of that. Why?
So this stuff isn't going to be on exam 1?
Yes, it will be.
Oh, okay.
Cool. Yeah, I don't know why you get hyperreflexia with an upper motor neural lesion. So I'll look into that. I'll get back to you with a good answer later on. So this is definitely going to be on your first exam, especially with the actual signals here. So we'll talk about these signals in a little second. So what's going on here? You're basically tapping the patella tendon just below the patella, and that's going to send afferent signals through your muscle spindles. And we're going to talk about muscle spindles later on. That's not going to be on your first exam. We're not going to talk about muscle spindles or the Golgi tendon, organ, and all that stuff. We're going to talk about that later on when we discuss motor and sensory circuits. But for now, just suffice it to say that we have muscle spindles that are going to be stimulated through the patella tendon reflex. And then that's going to send signals to the spinal cord. And those signals are going to be sent both to the nerves that innervate both the extensor muscles as well as the flexor muscles. Oh my gosh. Internet. This internet connection sucks. This is such a train wreck. I'm sorry about that. My internet, I think, took a hit. Are you guys with me still?
Yeah.
All right. Sorry about that. This is becoming a train wreck. I'm almost better. I'm taking tomorrow off, so I'll be there on Thursday. So we'll do our next lecture in person. I hate doing Zoom.
Are you taking tomorrow off, like for patho?
I'm actually taking tomorrow off, but I'm going to be doing Zoom again.
Oh, good.
Yeah.
Okay,
so
perfect.
yeah, perfect. I love doing
That's my
Zoom.
only class tomorrow, so I can stay at home.
Yeah, there you go. Hey, silver lining, right? It's going to suck because I hate doing Zoom lectures, but you know, you do what you do what you can. All right. So, do you guys see my screen again? My
Yes.
PowerPoint. All right, cool. So again, muscle spindles, that's going to be the sensory input, right? So whenever you tap the patella tendon, then you're going to stimulate your afferents, and that's going to be your muscle spindles. And then that's going to send signals to your spinal cord, and then you're going to have signals that are sent both to your flexor and extensor muscles. For the patella reflex specifically, you're going to have your extensor muscles are the ones that are going to be stimulated. That's why you have extension of your lower extremity with the patella reflex. Your interneuron is going to send inhibitory signals, okay? Interneuron is going to be specifically inhibitory in this reflex. And it's going to send inhibition to the flexor muscles. And so the flexor muscles are going to be turned off. That's why you only get extension. And what's the purpose of this? The purpose of this basically is to help maintain balance. So in a real life situation, when you're like walking along the road and maybe something like bangs onto your leg and it stimulates the afferents so you don't like fall over, this reflex basically helps you stay upright. It helps you stay on on both of your feet. Right. Later on, we're also going to talk about the extensor, like the cross extensor reflex. There's another reflex that happens, like if you step on something sharp or you have a pain stimulus and then you take your foot away, right? The foot that's affected gets taken away, but the opposite side gets stimulated. So now you have an extensor reflex so that you can still maintain your posture and not fall over when you have a reflex to basically step away from the painful stimuli. Now, if you were to record this using probes and sensors, this is what you're going to be seeing. You're going to see spikes. Now, this is the way that neurons communicate, right? Neurons are going to communicate through spikes, and it basically looks like Morse code. So spikes, so here's a really good definitions. Neurons communicate with each other with discrete electrical signals called spikes, and they work in continuous time. And here's another quote from a really great book by Bentov. Roy, you're the one who suggested that I read this book.
Excited for this, and now it's cutting out.
Oh no, is it cutting out?
Yeah.
It did for a second, but you're back.
Okay, cool. So this is the way that neurons are communicating with each other, is through spikes. And so it kind of sounds like a Morse code. And I put a couple videos in here that basically do a comparison between Morse code, right? Like, for example, SOS, right? So this Morse code over here was from, I think, a ship that was being struck during World War II. And then compare that with neuronal spikes. So play that video later on your own time so you can hear kind of the similar sounds that come from communication through Morse code versus the spikes on a neuron. So this is what Bentov said. In short, our senses translate the surrounding reality to us into a Morse code language of action and rest. Action comes when the neurons fire its spike, and rest comes as the cell is regenerating and readying itself for the next firing. And so now, with the myotactic reflex, this is another thing that I definitely want you to be familiar with too. So if you were to visualize the spikes during neuronal firing, make sure you know what it looks like when they're either being activated or if they're being inhibited. So here's your patellar reflex. So you have the hammer tap on the patella tendon. Your afferent sensory fibers are going to be increasing the frequency of spikes, not the amplitude. Right? Amplitude would mean like you have a larger amount of signal coming in, but no, for neurons, it's specifically the frequency. So the faster the signals are being sent, that means that you have a higher frequency of signals. You have like a higher concentration of spikes that are going to be taking place in a shorter period of time. Now, that's going to be sending signals to your motor efferent extensors, right? So your motor neurons are going to be now causing the extensor muscles, like your rectus, you have your rectus femoris, you got your vastus medialis, vastus lateralis, vastus intermedius. All those muscles are going to be now causing extension of the muscle. Then you have your interneuron. And remember, your interneuron is going to be specifically inhibitory in this case. And so look, your interneuron is also firing at a very high rate. And because it's inhibitory, now it's going to send signals to the flexor muscles to basically turn them off. And that's why you get a reduction of signals in your flexor muscles. And so if you were seeing this chart, make sure that you are able to interpret what the spikes represent. Right? You have a higher frequency of signals that are going to be stimulating the muscles involved in the myotactic reflex. Any questions on that? All right, cool. So here's another image. Well, this image is actually not from your textbook, but I pulled this image to basically demonstrate the different layers of the cerebral cortex. We're going to go into this later on, but you have 6 different layers of the cerebral cortex, and all they're going to be involved in different types of processing. So this is just basically FYI for now. I'm not going to test you on this just yet. This will show up later on in this semester. But let's just kind of briefly go over each of these different layers. So in your outermost layer of the cerebral cortex, just below the pia, right, when you see pial surface, that means that's the pia mater that surrounds the brain. Right below that, you have the layer 1 molecular layer. That's going to be mostly consisting of dendrites, as well as axons. And those are going to be receiving signals, and they're going to be sending signals down into the deeper layers of the cortex. Then you're going to have layer 2. That's going to be a granular layer. When you hear the term granular, it's literally just looks like little granules if you're looking at it on a light microscope slide. So it's going to look just like a bunch of granules. It also receives inputs from the outer layers of the cortex. And then just below that, you're going to have your pyramidal layer, Why is it called the pyramidal layer? It's because these cells literally look like little tiny pyramids, right? So they're going to be triangular shaped, and so that's where you get the term pyramidal layer. These are also going to be receiving signals from the higher cortical regions. And then as you get a little bit lower, you get another granular layer. And again, looks kind of like granules when you look at it on a light microscope. So that's going to be this region right here. So it looks like more granules. And then you have your inner pyramidal layer. And again, these neuron cell bodies look like pyramids. These are going to be very much involved in like motor movements. So that's going to be heavily implicated in motor function. And then as you get to the lower layers, before you get to the white matter, which is going to be your neuronal tracts made up of axons and myelin sheaths, that's where you have multiform layers. So it's got different types of shapes of the cells, and that's where you get lots of integration of information, and that's going to be sent into the outputs that information into the thalamus. And so remember, the thalamus is going to be one of the major relaying points for all sorts of different information, sensory and motor information. Again, we're going to talk about these 6 different layers later on this semester, but I'm not going to hold you accountable for that just yet. We're going to revisit this later when we talk about motor and sensory input of the cells. All right, now let's talk about a couple different types of other types of visualization methods for neurons in neuronal studies. So you can either use genetically engineered viruses. That's going to help you look at the presynaptic versus the postsynaptic cells. So you have a genetically engineered virus that can allow you to visualize the way that cells are able to communicate with each other, presynaptic and postsynaptic. And then there's another thing that you can use. It's called calcium imaging. And so you're going to be using calcium type dyes to be able to visualize neurons as they're firing under different circumstances. And so here's where you're using genetically engineered viruses to be able to visualize different types of cells. So remember, we're like looking back at the different cortical layers. So this image specifically is looking at the second layer. So that's going to be looking at this layer right here, the external granule layer of the cells. And so they're just going to be looking like little tiny dots. Basically, these are going to be like little tiny granules. As you would go deeper into the layers of the cortex, then you would start seeing, for example, pyramidal type cells that look like little pyramids or triangles. So that's using genetically engineered viruses. And the virus, basically, you're able to illuminate it using green reporters. And that's how you get the activation of the virus to be able to actually see what these neurons look like under microscope. And here's another cool technique using calcium dyes. And so this study, this is another study that's used in your textbook to highlight how calcium imaging works. And so basically what you do with this specific type of study is that you have an opening within the cranium. And so it's going to be targeting specific regions within the occipital lobe and the visual cortex to be able to see the cells as they're firing depending on what kind of sensory input that they're receiving. And so with this specific study, they had the mice look at different orientations on a grid. And the grids had different angles of orientation. Some were like 0 degrees, some were at 90 degrees, some were at 45 degrees, etc. And depending on which grid the mouse was looking at, different cells within the visual cortex would actually fire. And they would be able to visualize this using calcium-sensitive dyes. And so here's the orientation of the grids. For example, you had some that were at 0 degrees, some were at 90 degrees, and so on. And then you had different cell types that were labeled as 1, 2, 3, and 4. Different cell types were responding to the different orientations of the grids in different ways. So for example, one specific cell might prefer to fire when looking at the grid at 0 degrees. Or one specific cell would preferentially fire at 90 degrees. And the image that's from this study is demonstrated here. You have your orientation, for example, at 0 degrees. Then you had another orientation at 45 degrees, 90 degrees, etc. And depending on which orientation you saw on the grid, or sorry, the mice saw on the grid, different cells in the visual cortex would fire. And this was all done using calcium-sensitive dyes. So super cool technique. It's invasive, of course. You have to, surgically remove a portion of the cranium to allow for the objective lens, the calcium sensors to be able to detect which neurons were firing in the brain, depending on which orientation of the grid the mice was looking at. Here's a couple of videos that basically talk about calcium imaging of cortical neurons. Go ahead and watch those videos on your own time so they can get a better idea of how calcium imaging is done in the lab. So super cool. And here's another really cool technique that's also highlighted in your textbook, using optogenetics. Now, optogenetics is really cool because it's going to be using genes that are going to be using different types of organisms' opsins, for example. These are different proteins that are light sensitive, and they're introducing them into the neurons of cells so that the neurons can actually get activated from different frequencies of light. And they're going to be using laser beams to be able to target different frequencies of light so that it can activate or inactivate different types of neurons. And it's going to be using, for example, bacterial opsins. So here's an example of the different types of bacterial opsins. These are going to be using these 7 transmembrane type opsins, and they respond to different types of colors, different types of color frequencies. And depending on what type of color frequency is being shined onto the cell, it's going to be opening different types of channels. And so when you have opening of different types of channels, you have different types of cells being activated, and you can actually use optogenetics to turn on or turn off different cells depending on what your neuronal circuit's involved. So here's an example using the striatum. So later on, we're going to talk again about the basal ganglia, but this is a study that basically is observing different cell types and different cell groups within the basal ganglia to see which ones get turned on and which ones get turned off, depending on the signal, the excitation signal being sent. So, for example, if you're illuminating certain cells within the striatum, those cells are going to be turned off versus turning on the same exact cells within the substantia nigra. Those cells might actually get turned off as a consequence. Now we're going to talk about the nigrocidal pathway later on, but the substantia nigra is what produces dopamine. This is especially important for initiation of movement when we're talking about the direct pathway, the D1 pathway. So you have dopamine being sent to the striatum, and then when the striatum can get activated, it's thereby going to inhibit the globus pallidus, which is another region of the brain that's basically the off switch, and the off switch is always on, right? It's always turning things off until it gets turned off. So once the globus pallidus gets turned off, then you can actually initiate motor movement. So this optogenetics study was actually kind of cool to be able to demonstrate how stimulating certain regions of the brain actually help to turn on or turn off different areas of the basal ganglia. So here's another slide talking about that. So you have lasers again.
I have a question.
Yes.
Wait, what is D1 and D2 again? Are they just pathways, or are they? Because I know they have to do with the basal.
Right. Yeah. D1 pathway is called the direct pathway, and when you have stimulation of the D1 pathway, that's what allows for the initiation of movement.
Okay.
So in order to actually, when you're conjuring up an idea to move whatever, like an appendage, or to maybe like lift your arm or whatever, that's going to be taking place in the motor association cortex, which sits basically right in front of the primary motor cortex, which is the precentral gyrus. So you're like thinking, you're conjuring up the idea to move, right? And then that's going to send signals to the D1 pathway to allow the initiation of movement. To allow for the movement to take place, you actually have to turn off a certain region of the brain called the globus pallidus. Once you turn that region off, then you can actually initiate movement. Until you turn that portion off, you can't have movement. And then the D2 pathway is going to be the opposite. It's going to be inhibition of unwanted movements. And so D1 pathway and the D2 pathway both involve the substantia nigra and the striatum, which we'll talk about later on. So here's where optogenetics comes in with this particular study. So when you shine the light on this region of the striatum and you record that area, the striatum basically gets turned on. Whereas if you shine the light on the same area within the striatum, then the substantia nigra basically gets turned off. Right? So different areas that get stimulated, for example, with optogenetics, using a light to be able to stimulate those different opsins on the cells. By stimulating those areas, some neurocircuits might get turned on, but some neurocircuits might get turned off, which demonstrates that there are excitatory neurotransmitters and neurons, as well as inhibitory neurotransmitters and neurons. For example, the areas that get turned off within within the basal ganglia, when they get turned off, it's going to be through GABA receptors, which we're going to talk about later. But GABA is going to be one of the major inhibitory neurotransmitters. Whenever GABA gets released, that's going to tell neurons to basically stop, to reduce their neuronal firing. On the other hand, one of the major excitatory neurotransmitters is going to be glutamate. Whenever you release glutamate, that's going to tell your neurons and the associated neural circuits to basically turn on, right? So it's going to be an excitatory type of neurotransmitter. And again, we're going to be discussing those neurotransmitters later on when we talk about neuronal signaling. And here's the example of optogenetics basically allowing for that section within the substantia nigra to be turned off. So as soon as you stimulate the substantia nigra, the striatum, in this example, the substantia nigra basically gets shut off as a consequence. So that's optogenetics in a nutshell. Just make sure that you're familiar with the fact that it's going to be involving opsins that come from different types of organisms like bacteria. Those get integrated into the cell membrane. And then when they get stimulated by a certain frequency of light, that's effectively going to turn on the cell to create a cascade of neuronal signals. So that's the idea behind optogenetics. So in short, or in summary, what we just talked about, we were just discussing neuronal systems. Neural systems basically involve neuronal circuits and how they're basically organized to perform different types of functions. You also have unity of function, where you have different regions of the brain that are going to be responsible for specific types of actions, right? And then we discussed already things like the precentral and postcentral gyri, which had, the homunculus, which shows that you have different topographical areas in the brain that are going to be responsible for different functions as well. So in the motor cortex, for example, you have regions that are going to be responsible for movement of your hands, movements of the face, and then the sensory input in the postcentral gyrus. You're going to have regions that are associated with the face and different parts of the body to basically integrate different types of sensory input. Other ways that the brain and neuronal circuits are studied, lesion studies. So we talked about Phineas Gage already. So he was one of the first famous examples of using a lesion to be able to see what the actual function of the area affected is, right? So for Phineas Gage's case, you had that railroad spike going right through his frontal lobe. And at least for a period of time, he had frontal lobe damage, which caused him to be very erratic, very impulsive, very crude, vulgar, etc. And again, he did return to to baseline over time. But that's an example of a lesion study. Okay, so you see how a certain area, if it gets damaged, what would be the actual clinical manifestation of the damage, right? So lesion study that affected Broca's area, for example, that's where you start seeing that a person might start getting blunting of speech, right? They're going to have difficulty in forming actual words, versus lesion to Wernicke's area would cause an individual to have a difficult time interpreting words, and they would speak in quote-unquote word salad. There's other things that you can do to be able to study different parts of the brain. So you can use transgenic reporters. This is basically looking for specific type of genes. And so effectively you're seeing what genes are going to be responsible for different types of proteins. And you can basically look at a specific gene, a specific protein, and basically isolate that in comparison to all the other genes that are being encoded by, different proteins being encoded by different genes. You can also use antibody labeling, right? Use an antibody to be able to label specific proteins. You can also use that with like immunofluorescence, for example, to be able to, make different proteins light up underneath the microscope. You can also do things like in situ hybridization. Effectively, you're looking specifically for different types of mRNAs. So you're labeling DNAs as well as RNAs, and being able to see how those proteins and also tissues are affected by different types of genetic mutations, for example. So we're going to talk about Drosophila later on when we discuss, for example, embryonic development of different parts of the body, right? A lot of the genetic transcription factors and genetic markers that you see in animals like Drosophila, for example, can also be seen as well in mammals such as ourselves. So we'll talk about that later on. So super cool ways to study the neuronal systems. Here are other examples from your textbook that talk about different types of staining techniques. So you can use, for example, radioactive amino acids. You can use fluorescent stains. So make sure you know what your fluorescent stains look like. Those are probably going to show up on your exam. Here are other types of stains. This is looking specifically at the dorsal root ganglia, using different types of tracers. This image over here is looking at different types of proteins that respond, for example, to cholera toxins. That's what's stained here in green. So those are just different examples from your textbook. Here are some other examples from your textbook as well. These are transgenic reporters. So not super high yield. I'm not going to test you on most of these. I'm just going to test you on the major staining techniques that we talked about earlier. For example, Golgi stains, fluorescent stains, etc. All right. Any questions on any of that? All right, cool. So here's another way that you can study different regions of the brain using things like probes and stimulating different neurons in the brain and seeing how those, when you stimulate different neurons in the brain, how that results in some sort of effect in the body, for example, like motor neuron stimulation. And so receptive fields. So this is where you can have stimulation of a part of the brain and seeing how the receptive field gets affected in, for example, primate models. We're going to talk about how they can use, monkey models, for example, to be able to study how neuronal circuits come into play. And they use single unit recording. So definitely watch this video when you get a chance after we're done. That gives you a nice summary of what single unit recording looks like. So this is, again, from your textbook. So this is your somatosensory cortex, right? Part of your parietal lobe. That's going to be what integrates a lot of sensory inputs into your brain. Now, if you were to record regions of your somatosensory cortex, you'd be able to see how stimulation of different parts of your body would be recorded in the brain. So in this example, again, they're using primate models to be able to study these neuronal circuits, where they're going to be stimulating a certain area of the body. So for this example, they're using the arm. And so they're stimulating the center of the receptive field. And then for the area around the receptive field, this is called the surround. That's the technical term for this. The area within the surround is basically going to be turned off. So it allows for your brain to focus in on the area that's actually being stimulated. So in other words, this center region within the receptive field is the area that's going to be most stimulated, resulting in the most neuronal firing within the somatosensory cortex. Whereas the surround is actually going to be inhibited. So you have reduced neuronal firing within the surround, and you have increased neuronal firing within the central receptive field. And there's going to be an image here on the right side that also demonstrates that. So here's where you have maximum amount of sensory input, right? That's where you have higher frequency of neuronal firing, versus within the surround, you have decreased neuronal firing, right? So that basically allows for your brain to focus in, to hone in on the regions that are actually being stimulated, so that I can try to basically ignore the areas that don't need as much attention. Right, so it's kind of like a filtering. It effectively acts as a filtering mechanism to filter out unnecessary information. Any questions on single unit recording and the surround and central field and all that? All right, cool. So let's move on. Let's talk again about different regions of your nervous system. So we talked already about central nervous system, right? Brain and spinal cord versus your peripheral nervous system. Make sure that you're aware that central nervous system is going to be associated with efferent signals. So you have efferent signals being sent out to the rest of your body, versus peripheral nervous system is going to be mostly associated with afferent signals. Signals are going to be coming in to your central nervous system, mostly sensory input. And so in summary, you have your external environment. When we say internal, we're also talking about, for example, your enteric system, right? So you have a lot of nerves coming in from your GI tract, as an example, that's going to be receiving information from your internal organs. As well as, for example, like baroreceptors within your vascular system, right? You have, you have your baroreceptors in your aortic arch. You have baroreceptors within your carotids, right? Whenever your blood pressure gets increased, that's going to be sending afferent signals to your central nervous system to be able to now result in a parasympathetic activation, right? Parasympathetic response to slow down your heart rate as a consequence. So that would be an example of internal stimuli resulting in afferent sensory signals. External, pretty straightforward, right? Like, think about changes in temperature or pain stimuli. Any sort of external environment signal is going to be resulting in a central nervous system response. For, changes in temperature, if it's too cold, you're going to get signals from your hypothalamus now to result in, for example, shivering. If you're getting pain signals, then you're going to get a motor response to get away from whatever's causing the pain stimulus. Then you have your sensory input coming in to your central nervous system through your going into your cerebral hemispheres, for example, your diencephalon, which consists of your thalamus and your hypothalamus. Then you also have coordination of movement through your cerebellum, and then have input going down into your spinal cord, and then eventually you have your motor components. And then that's going to be subdivided into both visceral and somatic. When we talk about somatic, we're talking about body, right? So soma means body. So when you have a somatic response, that's going to be sending signals to your skeletal muscle. So you can initiate movement, as an example. Versus visceral, that's going to be your autonomic nervous system, so sympathetic, parasympathetic nervous system. It could also involve enteric divisions. When we say enteric, we're talking about your GI tract, right? Your mesentery, for example, that's all within your GI tract. And so enteric division is going to allow for, you guys tell me actually, when you have parasympathetic stimulation of the enteric nervous system, what is that going to be doing to your GI tract?
Isn't it rest and digest?
Yeah, rest and digest. What kind of motor signals are going to be sent to your GI tract? What's your smooth muscle going to do?
Would it be peristalsis?
Yeah, exactly. You're going to start getting peristaltic contractions, peristalsis, and it's going to start to allow for movement of chyme, which is the food that you consume, to move throughout your GI tract. And so that would be part of your enteric division. So again, smooth muscles, right? So peristalsis, that's all involving smooth muscles. Again, smooth muscles can also be seen in your blood vessels. And so if you have parasympathetic activation of the blood vessels in your GI, what are they going to do?
Dilate.
Yeah, they're going to vasodilate. So when you're consuming a meal, you're digesting it, you're going to be absorbing nutrients, you're going to have vasodilations of all the vessels within your mesenteric system. So you can absorb nutrients. If you have a parasympathetic response, what's going to happen to your blood vessels in your peripheral circulatory system?
Are they going to constrict?
Yeah, you're going to have vasoconstriction. And then we're going to talk about this later on with all the neurotransmitters and what they do. If you have epinephrine, norepinephrine released from your adrenal glands, adrenal medulla, that's going to cause vasoconstriction of your circulatory system. Cardiac muscle is also going to be heavily involved in that too, right? Because that's all part of your autonomic nervous system. Cardiac muscle in a parasympathetic state, it's going to cause bradycardia. It's going to slow down. In a sympathetic state, it's going to cause tachycardia, right? Yes, what's the question?
Okay, so this probably is not neuro much, but I was wondering if, you know how whenever you lift weights and they're like, okay, now go smash a protein shake or like smash a good old chicken breast. But aren't we, like, we're not resting and digesting. We just finished a workout. So isn't our body still in fight or flight? So
after eating a meal, you will definitely go into a parasympathetic state. Absolutely.
Even if you just completed like an intense workout.
Yeah, I mean, eventually your body is going to switch gears, right? Especially after consuming a meal. Like after you eat a meal, your body's going to switch gears because now it's going to have to go into a digestive state, right? You're going to have to go into a parasympathetic state to allow for the food to be digested properly. So yeah, your autonomic nervous system will definitely switch over.
Interesting.
Which is one of the reasons why it's a bad idea to go swimming after you consume a large meal, right? If you go swimming after you eat a large meal, you're supposed to be in a parasympathetic state, but now you're trying to go swim, which is, you have to like basically use your skeletal muscles, and then that's why you start getting cramps and things like that. Blood starts getting shunted into wrong places, and so that's a huge concern with swimming after eating. So that's one of the things that's going on there. So that's a really good question. But yeah, you would definitely switch gears. How long does it take? I'm not exactly too sure, but I'd imagine that it would vary from individual to individual, but you would definitely see your body switching gears there. So anyways, visceral motor neurons. Visceral motor neurons, that's going to be part of your GI tract, for example. It's going to be part of your heart, cardiovascular system, respiratory system, everything that's within your heart, lungs, etc. I take that back with GI tract. That's going to be more of your enteric division. So enteric division is going to be GI tract. So that's going to be your rest and digest, for example. Visceral motor neurons, though, that's going to be part of your heart, your lungs, things like that. And then again, you have sympathetic nervous division, and then you have your parasympathetic division as well. Now, in terms of where these nerve fibers come out of, it's good to know that the sympathetic division is going to be coming off of the sympathetic trunk, and that's specifically at the thoracic and lumbar regions, which I'm going to show you in a second, versus the parasympathetic division that specifically comes off of your cranial as well as your sacral levels. So let me show you this image here, and that will demonstrate that a lot better. So here's your sympathetic trunk. So it's going to be coming off of your thoracic, the thoracic levels of your spinal cord. Okay, so thoracic as well as lumbar. And so here are your sympathetic ganglia. Right, those are going to be all of the neurons involved within the sympathetic trunk, and then we're going to get into this more when we get into like synaptic, connection. We're going to talk about the different neurotransmitters involved, but just to briefly introduce the neurotransmitters, we're going to get acetylcholine as part of your preganglionic fibers, and then for the sympathetic division, you're specifically going to get your catecholamines as part of your postsynaptic fibers. So norepinephrine, epinephrine, those are going to be released by the postsynaptic fibers. Acetylcholine is going to be involved across the board as your presynaptic neurotransmitter for both sympathetic and parasympathetic. But when we get into the postsynaptic neurons, it's specifically going to be your catecholamines when we're discussing sympathetic nervous system. Again, though, parasympathetic nervous system, you're going to have cranial division. That's where you have the nerve fibers coming off. And then your sacral levels as well. So it's going to be cranial and it's going to be sacral. Again, just to introduce the neurotransmitters, acetylcholine is going to be involved in both pre-ganglionic and postganglionic nerve fibers. Okay, so the similarity is acetylcholine is associated with your preganglionic for both sympathetic and parasympathetic, but when you get into your postganglionic fibers, it's going to be catecholamines for sympathetic and acetylcholine for your parasympathetic fibers. And these are the different, things that these divisions do. So with sympathetic, they're going to be involved in fight and flight, right? Increased metabolic rate, increased respiratory rate, increased heart rate. We're going to talk later on about the receptors, but it's good to be familiar with these. Beta 2, you have two lungs. Beta 2 receptors allow for bronchodilation, right? What's a drug that allows for bronchodilation of the respiratory tract? Any of you have asthma? Yeah, albuterol, exactly. So albuterol is going to be a beta 2 receptor agonist. Beta 1, I like to think of you as having one heart, right? We have one heart. Beta 1 receptor activation is going to cause tachycardia, elevation of heart rate, increased contractility, etc. What's a class of drug that blocks these receptors? That would be used for patients with like hypertension, as an example. Any guesses? You can guess just by looking at the receptor here. It's the class of drug. The receptor is actually in the name of the class of drug. You guys familiar with that?
Is it just called a beta blocker?
Yeah, beta blockers. That's what I wrote right here. Can you guys see my screen still? So yeah, beta blockers, exactly. So metoprolol, atenolol, labetalol, all the LOL drugs. Those are your beta blockers, and they basically block sympathetic activation of the heart, right? So it's going to cause your heart to now go a little slower. That's what beta blockers do, effectively. And then for parasympathetic activation, you get everything is the opposite, right? You get decreased metabolic rate. Your heart rate goes down. Your blood pressure goes down. However, you get increased secretion of your digestive glands, your salivary glands, right? You also get defecation and urination being stimulated by the parasympathetic nervous system, right? That's going to be kind of part of this whole entire sacral division as well. So here's a summary of parasympathetic versus sympathetic nervous system. With parasympathetic, if you were in a rest and digest mode, your pupils would constrict, right? Versus if you were being chased by a lion, you want to get as much information from your environment as possible so you can process as much information, so your pupils would dilate, right? Sympathetic, heart rate slows down. Parasympathetic, your heart rate goes up. Reduced salivation, right? There's certain medications, like if any of you guys take drugs like Vyvanse or Adderall, right? If you have ADHD, those kinds of drugs are going to be causing a sympathetic activation, right? It's going to cause decreased salivation. It might cause a little bit of dry mouth. There are certain drugs that can do the opposite, that can increase salivation. Anyways, so here's just a good chart that basically summarizes those major divisions, parasympathetic versus sympathetic. Other things that I want you to be familiar with too. So we already discussed gray matter versus white matter, but nuclei, whenever you hear the term nucleus, that's going to be a collection of neurons, and they all have the same type of structure and they have the same type of function. So you're going to have things like the subthalamic nuclei. Those are going to be different types of nuclei that basically sit below the thalamus that are involved in all sorts of different types of functions. But they're going to be all similar types of neurons that all do similar types of things. And then you're going to, also within your brainstem, you're going to have different types of nuclei associated with the different cranial nerves, right? I'm not making you guys remember every single one of those nuclei, but you have nuclei that are going to be associated with cranial nerve 7. You have nuclei associated with cranial nerve 8, right? The vestibular as well as the cochlear nuclei. If you're outside of the central nervous system, nuclei are going to be referred to as ganglia. So we talked about the the dorsal column ganglia, right? The dorsal root column. Those are going to be associated with a bunch of neurons that basically function in a similar type of, do a similar type of function, right? They're going to be associated with, for example, sensory input, right? So ganglia are the nuclei of the peripheral nervous system. But when we talk about a bunch of neurons doing a similar function within the central nervous system, we refer to it as a nucleus or a nuclei. The white matter, again, those are tracts, right? The reason why it's white is because it's made up of a lot of myelin, right? All this area right here, that's all myelin sheets, right? And so you have axons covered in myelin sheets, and they're going to be allowing for communication with areas far away in the nervous system, right? So you have commissures, right, which allow for the communication between your different cerebral hemispheres, right and left cerebral hemisphere. Great example of a commissure is going to be your corpus callosum. And then you also have your columns, which we already talked about. You have your spinothalamic tract, you have your cortical spinal tracts. Those are all referred to as columns. That's the umbrella term. All right, now let's switch gears and talk a little bit about genetics. I did say before that this is not a genetics class, but unfortunately there's going to be quite a bit of genetics, especially coming up when we discuss embryogenesis and embryologic development and whatnot. There's several genes that are going to be involved in that, including transcription factors and things like that. But let's revisit basic biology. The central dogma of genetics or biology is going to be genes, which make up DNA, get transcribed, right, into RNA, which get translated into proteins. Now, when we talk about genomics, we're going to be basically talking about the entire genetic sequence involving all the DNA, both in species as well as an individual. So it's not just an individual, it could be also across an entire species. And then we also study mutations, right? How different types of mutations of DNA can have massive effects on, for example, brain development. Right? We're going to talk about things that might cause, for example, conditions like lissencephaly, which is a smooth brain disorder, We're going to talk about holoprosencephaly later on as well, which is where you have deficiency in like the lobe development, right? So instead of having two hemispheres, you might have one hemisphere. So it's changes in like the migration patterns of different types of cell structures and neuronal circuits. So we're going to talk about a lot of that later on when we get into development next time we meet. So the human genome, we have lots of genes within the human genome. It's going to be 20,000 genes. This is a really cool diagram from your textbook. So it talks about all the genes in your body. So you got 20,000 genes. Look how many genes are actually dedicated to the nervous system. 17,000 of your 20,000 genes are all dedicated to your nervous system. That's absolutely insane. So massive amount of genes are associated with your nervous system. And of those genes, 6,500 are going to be associated with the brain alone. So that means 30% of all of your neurons are associated with the brain. Oh boy. Okay, sorry. This will be the last time I lose you, I promise. We only have like 10 more minutes left anyways.
It's a sign.
It's a sign. It's a sign that I can't take any more sick days. Where did I lose you guys?
Right when you were going to say how cool it is that the genes are expressed in the CNS.
Oh, okay, yeah. So, cool thing about your entire genome, like a huge majority of your genes are dedicated specifically to your nervous system. Massive amounts. So 20,000 genes total in your entire body. The vast majority, 17,000 of which are going to be dedicated to your nervous system. 6,500 are dedicated to the brain alone. So like 30% of all of your genes are dedicated to your brain. So absolutely insane how much of your entire genome is dedicated to your nervous system. So super cool. Here's just an image basically depicting single gene type expression in your brain and how you could visualize that with different types of neuronal studies. Here's another way that you can study how genetic variation or genetic mutations can result in different types of disorders. So here's a abnormality associating the spindle-like microcephaly associated gene. So this is going to be called ASPM gene, and this causes microcephaly. Microcephaly just means that you basically have a tiny brain. There's another condition that a person is born with no brain that can also be associated with a genetic disorder that's called anencephaly. So microcephaly, small brain, anencephaly means no brain. So the person literally has no brain. So a scary condition that can also be due to genetic mutations. Other things that you can see with genetics in terms of like studies, you can do splicing. So genetic splicing, you can do splicing variants. So here's an image that I pulled. This is not from your textbook, but this is how you can do different types of splicing studies. So consecutive splicing means that every single portion of the gene is actually going to be transcribed and translated, versus other types of splicing techniques where you can remove, for example, exons. You can also retain introns. And by the way, make sure you know the difference between exons and introns. Which one of these two are going to actually be transcribed and translated? Is it exons or is it introns?
Exons.
Exons, yeah, good. So introns are, it's a misnomer to call them junk DNA or garbage DNA because, there's a lot of evidence that suggests that introns are involved in regulation of, transcription and translation. So they can still serve a purpose. But exons are the ones that are specifically turned into actual proteins. That's like the final product is going to be coming from your exons. And so you can do different types of splicing techniques in different genetic type studies. Speaking of genetics, make sure that you're familiar with some basic genetic terminology. So make sure you know what genotype is versus phenotype. Your genotype is basically what type of alleles you have. Your phenotype is the way that your alleles are going to be represented physically, right? That's going to be the physical manifestation of your genotype. Make sure you're familiar with homozygous versus heterozygous, dominant versus recessive, right? So if a person, for example, is heterozygous and they have for example, let's talk about like sickle cell anemia. Sickle cell is like a homozygous type condition. You have it's a homozygous recessive condition. If you're heterozygous, does that mean that you're going to have the disease?
You can be a carrier of the disease, but it can be dormant later in life, but you don't have it. That's the whole thing.
Yeah, so phenotypically speaking, you're not going to have the disease, but you are a carrier, right? So if you're heterozygous, you're a carrier of the recessive trait. But if you are homozygous for the recessive allele, then you're going to be a carrier of the disease. Versus a dominant disease, even if you're heterozygote for the dominant allele, you're going to carry the disease, right? Because that dominant allele is the one that's going to be phenotypically expressed. These are other ways that you can study genomics. You can do homozygosity mapping. You're looking at multiple different genes associated with a disease. You can also look at the entire genome, so that's genome-wide association studies. You can look at inheritance patterns for single types of genes using those types of studies. And then another thing that I want you guys to be familiar with, so actually these ones are not super high yield. Make sure you know just basic genetic terminology here. I'm not going to test you on homozygosity mapping or genome-wide association studies, but you might see something like this: transgenic animals versus knock-in and knock-out animals. What is a transgenic animal? A transgenic animal is an animal that has a portion of a genome that was taken from another organism and it was introduced into the genome of that animal. And that can be used for multiple different types of studies. So I'll give you one example of a proposal that we're actually putting together right now for a study that we're doing for Parkinson's research. One of our models is that we're going to be using transgenic mice to be able to test for different drugs that might help to prevent, for example, the symptoms and severity of symptoms in Parkinson's disease. So if you had a transgenic mice that would express a certain type of gene, then you can use that transgenic mice or model to be able to study the disease. So the transgenic mice that we're using have a heightened expression of an alpha-synuclein mutation. Alpha-synuclein is one of the hallmarks of Parkinson's disease. If you have a transgenic rodent that has a greater expression of that gene, then you can actually induce Parkinsonism much easier on that mouse than you could on a non-transgenic rodent. And so if you use a transgenic rodent, for example, for Parkinson's disease, you can induce that using a compound called MPTP. I'm not going to test you on that. But if you induce Parkinson's disease on that rodent, it's going to be much more easier to induce that disease on a transgenic rodent than a non-transgenic rodent. And that gene was derived from human, right? It's basically a human alpha-synuclein gene that was introduced into the mouse genome. So that's kind of like a good illustration of how you would use genes from one organism to be introduced into another organism for the sake of genetic studies and studies of different diseases. So this image over here, they were able to actually introduce green fluorescent proteins into a mouse so that if you put it underneath the black light, now the mice that are here on the left and right side, those can both glow underneath a black light. The one in the middle here, that's a non-transgenic mouse. It does not have the green fluorescent protein. Knock-in and knock-out animals, that's where you basically insert or remove a gene. In other words, you can insert a gene to see if that gene actually causes any sort of like weird effects, in terms of like phenotypic gene expression, or you can remove the gene and see if that also has any sort of detrimental effects on the animal. So that's what knock-in versus knockout animals mean in terms of like genetic research. So here's another example from your textbook talking about knock-in and knockout, or introducing new genes, modifying the genome of a mouse. And on that note, where are we at in time? On that note, I think I want to continue here next time we meet. We're going to talk a little bit about homologous recombination, but CRISPR is going to be one of the major ones I want to focus on for this class. Any questions? Any questions whatsoever? Apologies for some of the technical difficulties. I appreciate your guys' patience in accommodating me as I try to recover from this respiratory issue.
Sorry, I had a question about the textbook. I got kicked out and it's saying that I need a redeem access code or like an access code in order to get onto it.
Did you try using that same access code that I gave you guys earlier on a couple weeks ago this semester?
The access code said that it wasn't valid for me.
Really? Has anybody else had a similar issue? So just you, Sasha?
Yeah.
All right. So what I want you to do then is to email me. Email me at randall.clarkson@gcu.edu. Send me an email with your issue, and then I'll get you in touch with the tech support at Oxford Insight. And so they'll hopefully be able to grant you access once again. So sorry about that. That is definitely annoying. So I apologize for that.
No problem.
But if nobody else has any comments or concerns, then let's call it a day. Again, thanks for your patience. I can't believe I got sick again, but I'm feeling a lot better. I'll be back on campus for sure on Thursday, so we won't have to do this Zoom nonsense.
Feel better. Thanks for the lecture.
I appreciate it. Thank you so much for the kind words.
Thanks, Dr.
Thank you.
All right, my pleasure, guys. Have a good one. Take care.
See you.
All right, bye-bye.